Welcome back to Plantopia. Plantopia is the plant health podcast of the American Phytopathological Society, and I'm your host, Matt Kasson. I'm an associate professor of forest pathology and mycology at West Virginia University. This is the 10th episode of Season 5, and today I'm excited to welcome Dr. Jim Westwood, professor of plant and environmental sciences in the School of Plant and Environmental Sciences at Virginia Polytechnic Institute and State University, Blacksburg, Virginia. Dr. Westwood received a BA in biology from Concordia College in Moorhead, Minnesota, an MS in plant physiology from the University of Minnesota in St. Paul, and a PhD in horticulture, weed science from Purdue University in West Lafayette, Indiana. He joined the Department of Plant Pathology, Physiology, and Weed Science at Virginia Tech in 1994 as a research associate and scientist before transitioning to an assistant professor position in 1999. With promotion to tenure in 2005 and full professor in 2012. Dr. Westwood's research program lies at the intersection of weeds, genome evolution, and parasitic plants. He focuses on parasitic plants, their novel structures, and mechanisms for information exchange with their hosts. Studying these interactions not only gives Dr. Westwood and others a better understanding of basic plant biology, but also helps improve techniques for applied vegetative management. He's published more than 65 peer-reviewed research papers, several book chapters, and has served as co-editor for 2 books. Dr. Westwood teaches courses in weed science and management, molecular biology for applied plant science, and weeds that shape our world. Over his long career, he has advised or co-advised more than a dozen graduate students, including 9 PhD students and over 60 undergraduate students, and over a dozen high school students and teachers. He is the recipient of numerous awards and honors, including serving as a Fulbright Scholar at INRA in France from 2019 through 2020. He is also the recipient of a Lifelong Achievement Award from the International Parasitic Plant Society and was recognized by Virginia Tech as a faculty member of national distinction in 2016. Jim, thank you so much for joining us today.
Thanks for having me.
You studied biology as an undergrad and then plant physiology for your master's, both in Minnesota. Are you from Minnesota?
I am from Minnesota. I'm from Minneapolis.
Okay. And what first got you interested in plants?
I don't know. For a long time, I wanted to be a wildlife biologist like everybody else. Go chase deer or wolves or whatever through the wilderness. But in college, I realized that plants just made sense to me. And something about the biology just grabbed me at that point, and I thought I could stick with that for a long time.
Yeah. So, I mean, your first degree in plants was plant physiology for your master's at the University of Minnesota. Now, when you were a biology undergrad, did you have a plant-heavy kind of education, or was there any formative experience there that got you interested?
I had an instructor in sophomore year, Introduction to Plant Science, and she's the one who did it for me, really, who made plants seem exciting and comprehensible. Yeah, I was never going to go pre-med. But again, that looked really interesting. But at that point, I didn't really know much about the scope of the world of plants. And so when I went to do my master's at the University of Minnesota, I was in the botany department there. And I was introduced to all these different departments like agronomy and horticulture and plant pathology there. And it really expanded my world of plant science. I started to get into thinking about weeds and ended up doing my master's degree on a little bit of a herbicide question. So that brought me to the practical world of plant science.
Yeah, and then you developed an interest during your master's, and then you wanted to further expand on that at Purdue.
Yeah, but the critical thing that happened in there, I went to the Peace Corps for a couple of years. And so I worked in agriculture in West Africa. And I was in the country of Mauritania, which is on that west coast, just south of Morocco, if you know your African geography. So there I was working in agricultural extension, and it really hit home to me there that weeds were, in fact, a huge problem and very difficult to get rid of in that system, where it was mostly hand labor, right? You were hand pulling weeds. So here I thought I would take my plant knowledge and really put it to good use.
That makes a lot of sense. And I'm sorry I missed that actually on your CV that you were in the Peace Corps. And That makes sense given the abundance and diversity of parasitic plants in Africa and its impact on agriculture there. So the connection is pretty strong.
Yeah.
So that's when you first encountered parasitic plants. It was in Africa during the
Well,
What?
I probably encountered them. I didn't know it at the time. And
Uh-huh.
it wasn't until I was at Purdue and I was getting close to the end of my degree in weed science. And I went to seminar and there was a guy from biochemistry talking about witchweed, striga. And he had been working on the germination signaling. And he started to talk about this huge weed problem in Africa, showing pictures of basically the villages I had just lived in. And that really hit home. I thought then that was probably the most exciting thing I'd heard. I saw a plant interaction and I realized that I probably could have been a plant pathologist in a different life because I really love the interactions between different organisms. And so this idea of a plant attacking another plant really got me excited about parasitic plants. And then I determined at that point that's what I was going to do. And so I went off to a postdoc to study parasitic plants.
Well, and for so long you were housed within a department of plant pathology and physiology and weed science, and it makes sense. There was a lot of crosstalk there. Someone who studied plant physiology and weed science, you also could fit in well with other plant pathologists.
It was a perfect description for me. That it had a long name for that department, but yeah, it encompassed all of my interests.
Now, our listeners may not be super familiar with parasitic plants. They may have seen them, they might know one or two of them, but can you explain what are the defining features that constitute a parasitic plant?
So the big feature is the presence of an organ called the haustorium, which is unique to parasitic plants, and it's a special organ that the parasite uses to invade, make a connection, physical and physiological connection to the host plant. And actually, the term haustorium, many of your listeners will know from their studies of fungi and things, but the term haustorium was coined first for parasitic plants and then
Okay.
adopted in the world of pathology. But anyway, it's that organ that kind of defines the parasites that we study within this world of parasitic plant science.
And that more or less can be described as a nutrient exchange organ.
Yes.
And of course, like you said, we know in a lot of biotrophic fungi, biotrophic plant pathogens, haustoria are used to steal from the cells without bursting or piercing the inner membrane of the cell.
Functionally very similar. And the same thing. It's just withdrawing those host nutrients.
And similarly, for those listening, not all that different from arbuscules, which are the nutrient exchange sites of arbuscular mycorrhizal fungi that form beneficial partnerships with the roots of plants.
Yeah.
So we sometimes use terms like holoparasite or hemiparasite. What's the difference there?
Yeah, so that's relating to the level of dependence of the parasite on the host plant. Hemiparasite is going to be only partially dependent on that host plant for its nutrition. A holoparasite will absolutely require, let's say it'll take all of its nutrients from the host plant. So it may lack any green tissue or expanded leaves.
So holoparasites are typically achlorophyllous or lacking chlorophyll.
Right.
Whereas something like a leafy mistletoe would be a hemiparasite, but it would still be obligate because it's in the stem.
Yeah, so this is the obligate versus facultative. So the obligate has to have the host to reproduce. The facultative parasites are able to grow in the absence of a host, even if it's not quite as well as they would if they had a host.
So there's this, does it depend mostly on the host? Does it sometimes depend on the host? There's the obligate versus facultative. And then there's the whole, what part is being parasitized? And that kind of tells you a little bit of the relationship. So it could be a root parasite. It could be a shoot parasite.
Exactly. And so that, another way to divide things up. Yeah, what are they attacking? Is the root or the shoot? And then, of course, there's some parasites that will actually invade and grow within the host, like a mistletoe or the Rafflesia or something like that. That's mostly growing as like a thread of tissue within the host system.
Now, we tend to talk about these things as parasitic plants as like a single group, but we know that there's a dozen independent evolutionary lineages of
Yeah.
parasitic plants?
It's evolved in at least a dozen different families. So there's multiple different origins of parasitism. And yeah, I tell my students, it's not a fluke, it's not an accident. You get parasitism. It's this ability to steal from another organism instead of making food yourself is a time-honored strategy in the world of biology.
Absolutely. And I've read a stat that 1% of plants have a parasitic lifestyle.
Yeah, yeah. So maybe 4,000 species are parasitic.
Could you tell us a little bit about some of the main families of parasitic plants? I know you worked on a couple.
I can tell you about the main weedy species that we really care about, and that's the family Orbonaceae, which has probably the worst parasites in it. The witchweeds, the broom rapes, the strigas, and Orbonaceae that are attacking the roots of plants, and they are among the best studied parasites that we have. And another family would be the Convolvulaceae, Cuscuta family. So this is a family of vining stem parasites where there's nothing but a vining stem.
That's daughter.
Yeah.
Yeah. And then that's the morning glory family. That's
The morning glory family. Yeah. And then there's mistletoes, of course, which can be damaging to the trees that they're parasitizing. There are
So yeah, there's dwarf mistletoes and there's leafy mistletoes. Coming back to the broom rape family, I know around here we have a couple of commonly found parasite plants, Conifolus,
Yep.
bear corn, as it's
Yeah.
commonly referred to, and that causes cancer root on oak.
Yeah.
And there's the Epiphagus, the beech specific.
Yeah.
Yeah. So there's a number of broom rape family members present here in the eastern U.S.
Yeah. We do have representatives. Yeah. They tend to be growing on trees, but not in crop situations and not apparently doing a lot of damage. They're not like the really weedy species from this family tend to be over in Africa or the Mediterranean, Southern Europe, things like that.
I was in the Florida Keys a couple of years ago with my family, and I couldn't help but notice there's a lot of dodder as you drive down between Key Largo and Key West. That's a pretty ideal habitat for that?
Yeah, I don't know about that specifically, but I know when I travel to the south or to tropics, you tend to see it more. Yeah, so I think I like those warm climates. And I'm not sure which species it is all the time because those daughters are really tricky. There's not much different morphologically there to use to identify things. But we're getting better now with genetics.
Yeah. One of the parasitic plants, which is a unique or an oddball, these are an Ericaceae, the ghost pipe and pine sap, but they're something called mycoheterotrophs. They're parasitizing mycorrhizal fungi.
Right. I've talked to a few people who work on those. They don't often get included with the people who study parasitic plants because they're not directly attached to plants. These are plants, right? What you're talking about, the monotropa.
Yeah.
They're connected to a network of fungi, and they may be indirectly connected to other plants to get but it's not a direct parasitism of the other plants. But it's yet another, I think, fascinating lineage of plants. They figured out how to make their living from microorganisms.
Absolutely. And I know that's one that a lot of people bring up when we're out on forays and mushroom walks. A lot of people point to it because they think it's a fungus.
Yeah.
Because it's a chlorophyllus, and I have to explain to them, no, that's an angiosperm, and explain to them about parasitic plants. It's a great conversation starter, of course. Similarly, when I'm morel hunting in the spring, I always come up and see bear corn coming up, and they're like, "Oh, there's a bunch of morels." I'm like, "No, those are parasitic plants." So, what's really interesting and caught my eye about the broom rape family is they're important for several reasons. One is that they have a diversity of parasitic plants in them, but they fill the range of, like, dependent strategies, right? They go from holoparasites to hemiparasites, and there's also some free-living plants in there, right?
Yeah, they do. And what you just mentioned, that diversity of host dependence is what attracted me and some colleagues to them several years ago to design a big project to try to get at the genetic basis of parasitism. I'm working on these holoparasites, and a colleague was working on a hemiparasite, another one, a facultative hemiparasite. And we worked on what we call the Parasitic Plant Genome Project. And we're funded for, I think, 8 years or so to work on this. And at that time, it was transcriptome sequencing. We're trying to figure out what was the essence of that hostorium, right? How did it interact with the host, and what made a parasite? Could we trace back to the point of the origin of the hostorium, how it functioned, and maybe how a plant might then start to lose the capacity for photosynthesis and the genes involved in photosynthesis? And yeah, it was a very interesting project. I wish we could do it again now with all the genome tools where you could sequence stuff for real cheap, but we'll figure it out. It's pretty interesting.
Now, location is not the only important factor in describing parasitism. We talked about where in the plant, but like some tap into only the xylem, like Striga, and while others, like Daughter, they're tapping into both the xylem and phloem. And what determines that? When you're tapping into the phloem, presumably you're tapping into plant hormones, and you can alter plant hormone profiles, right? But is it more common to just steal water? Or is it more common to steal water and sugars and these things?
Really complex question. I think all parasites need the water. They're all after the water, and they all form direct connections to the host xylem that when you look at it in a scanning electron micrograph, it looks like a plumber's joint. You've got a T-joint there, where the xylem elements of the host are just intersected by the xylem of the parasite. So all of the parasites really want water. And then they'll take, in addition to that, I think they'll take whatever resources they can get. And this is still poorly characterized. There's a lot of room to figure out how this hostorium works. And there are some species for Orbancaceae where, to see even many years ago, I've shown micrographs where it looks like you've got sieve element continuity between the host and the parasite. But there you've got direct phloem connections. With something like Daughter, no one's shown that. And yet we've done a lot of work, and others have done a lot of work showing that there really is, the phloem continuity is very high. There's a lot of exchange going on between the Daughter and the host. And I think the parasite just has a way to get what it wants, regardless of the connection. Because even with the witchweeds, which don't have a symplastic or a phloem connection, it seems that they still get sugars from the host. And so they're probably inducing those hosts to dump sugars into the apoplast or something that they cannot by the parasite through the xylem connections. It's really interesting plant physiology. And how do you transport sugars and all the other nutrients, and then figure out how a parasite can get in and manipulate that system and hijack it so that the host is going to feed the parasite.
Yeah. In looking over some of your papers, I think my understanding of a hostorium from like the fungal plant pathogen side is a little bit simplistic. And in reading your stuff, I understand that your work and others have shown basically bidirectional movement of viruses, proteins, mRNAs. There's a lot more happening at that interface than we previously thought. And I'm sure you're discovering new stuff all the time. Is that what you continue to understand, that communication?
Yeah, what we've seen now evidence for the movement of all these macromolecules that you mentioned. And the challenge now is figuring out how it all works. So just to be more specific about it, we've seen that what we thought was the parasite was just like a vacuum, taking one unidirectional uptake of water and sugars and stuff. And then our work with the RNAs showed that it was actually an exchange. It's not quite equal, but there's a lot of parasite RNAs, mRNAs, microRNAs, and probably proteins as well, going from the parasite into the host. And at least some of those are serving as effectors, right? That we've got good evidence now that microRNAs are actually manipulating host genes. But this is also emerging as a theme with some fungal plant interactions and other interactions in plant pathology, where there's a kind of a battle of going on between these organisms, and proteins. In fact, it's not just proteins, but they can be microRNAs, and whatever the organisms can exchange can be used. I think the parasitic plants are also probably using hormones as well to sense and probably also to inject into the host as well. Plant has multiple tools it can use against another plant. Basically, they are speaking the same language.
That makes sense. You've worked on field bindweed, purple witchweed, Egyptian broomrape, dodder, sunflower broomrape. There's probably many more in there, but those were some of the ones that popped up in your list of publications. Those, or ones not mentioned, do you think present the biggest challenge to agriculture and plant health currently? I know you were talking about your time in the Peace Corps and a lot of these having an impact on crops in Africa. If you think about worldwide, which of these parasitic plants do you think are posing the greatest risk?
Yeah, good question. I think my standard answer is the witchweeds, guys, is the one that captures the most attention because it's so devastating to the cereal crops. So it's attacking corn and sorghum and millet and rice. In general, there's one species that attacks cowpea as well, but those main species are really attacking the staple crops in sub-Saharan Africa, right? So where you've already have many challenges to agricultural production, and here you've got this terrible weed impacting the crop as well. And so that's the one I think that most people would say that's the biggest problem in terms of human, if you could improve all that problem, you would improve human nutrition substantially in that region.
Now, Striga asiatica, I don't know, that's in the U.S., right?
That is in the U.S. Yeah, there's an infestation of that from the 1950s in the Carolinas, right on that border of North Carolina and South Carolina.
So you made a good point that parasitic plants are great at making seed. They do that well. And in my general plant pathology class, I teach about concepts of catch crops and trap crops, in particular as control strategies for Striga and other parasitic plants. Are those concepts that you generally know about and talk about? And can you tell the listeners what we mean by those things?
Yeah, those are common concepts in our field as well. So then the catch crop is a crop that you would plant that would be parasitized, and then you basically spray or you plow it under, kill the crop with the parasite as a way of depleting seed in the soil. The trap crop then is something that would trigger parasitism, trigger parasite germination, but not allow itself to be parasitized, call a false host or something. And so both methods would deplete the soil of the parasite seed. And so both have been kind of, the concepts are there. They turn out to not be so effective in the field. Just because of the longevity of the seeds of the parasite. And the example is like the broomrape in Israel, where it's a parasite of tomatoes. If you rotate out of tomatoes, you plant cotton, it can't be parasitized, it's not a host, but it does produce the stimulant to germinate the seeds. And you grow it for 20 years, and then you sell the land, and somebody comes in and plants tomato, and there's the parasite coming right back again. So on a real world scale, with the complexities of soil, it's just really hard to completely get rid of the seed.
Right.
Through that kind of technique.
Okay, so one other term I've heard is called suicidal germination with ethylene. Is that something that's done?
That's been the main mode of ridding the soil of Striga seeds in the Carolinas, where you can inject ethylene gas, which is somehow involved in the germination signaling pathway for Striga. Given the right conditions, so it's got to be good temperature and moisture for a few days. You get those conditions, you can go through with a burst of ethylene and trigger all germination without a host being present, and those plants will germinate and die, right? The seeds will germinate and die. And you can do that where you've got a little bit of money to invest and you're not worried about reinfestation from surrounding areas. So this has been an effective tool in the US for controlling small infestations, not work on a wide scale in Africa, where the problem is so big and the resources more limited.
While we're talking about kind of germination and germination stimulants. This was a question posed in one of your papers. Why would plants reveal themselves and allow for the germination of these parasitic plants? And, what you and others have learned over time is that really these signals are meant for arbuscular mycorrhizal fungi. And is it accurate to say those signaling pathways are being hijacked by parasitic plants?
Yeah, it's so the plants evolved this chemical signaling with vascular mycorrhizal fungi to facilitate colonization, especially when under nutrient poor conditions. And parasitic plants are able to detect the strigolactone compounds that are exuded by the host plants and detected by the parasite. And it's a great story in evolution, really, because you've got each kind of group of plants, family of plants, produces their own combination of strigolactones, right? These chemicals come in a lot of different variants, right? And so I'm not sure what good that does to the host plant, but for the parasite, they've then evolved a set of receptors. And the parasitic plants have multiple receptors that we propose are attuned to different strigolactones. And this is how witchweed seed, for example, can recognize a corn versus some broadleaf weed root growing by it. And it can recognize that host plant down to some level of specificity in order to germinate appropriately.
Yeah. Now, you said earlier, I think was a good point, is that the parasitic plants and their hosts, they speak the same language because they're both flowering plants, which brings us to an important point here that I hope you can clarify. So at some point, parasitic plants suppressed their own production of strigolactone so that they can sense it externally from their host in the environment.
Yeah, I wish I could provide more clarification on that. So we determined that the parasitic plants, in fact, do still make their own strigolactones. And so what's not clear completely is how it works and how they can pay attention to the external strigolactones without being confused by their own internal strigolactones. But I would guess it's either the type of strigolactones they produce. And we haven't, no one's, I think, yet isolated strigolactones from a parasitic plant. We just know that they have the genes and proteins to make them. It's the specificity either in the type of strigolactone, the timing they're produced, or the location which they're perceived.
Okay. And this is part of the larger conversation on like non-self recognition.
Yeah.
Because you don't want to be like, oh, there's a root, and you're actually parasitizing yourself.
Yeah, there's some good studies that being done with some of these facultative parasites. How do they recognize that they can recognize their own root? And some great work coming out of Japan on this question, I think, with an idea that there is a cell wall modification that allows the parasite to know the root next to it is another parasite or a host plant.
Yeah. Now, we were talking a little bit about control strategies, trap crops and catch crops. Of course, when we think about control strategies and integrated pest management, we often think about breeding for resistance. And I was reading a little bit about some of the pre and post attachment resistance you can have in plants that make them resistant to parasitic plants. Could you talk at all about any of those aspects because it's interesting that I think the context I read it in is that there were some maybe carrot varieties that were not susceptible to parasitic plants because of some either pre or post attachment resistance.
Yeah, so the pre-attachment resistance is the simplest, and it's what we've already been talking about. It's that signaling for the parasite seed germination, or that's the main one. People have realized it, so we know the strigolactone pathway, and so if you modify that or find plants that have low strigolactone exudation, you can have a type of resistance because the host plant then is invisible to the parasite. So that's all based on this kind of signaling. So the post-attachment resistance is less well-defined, and there's only a few cases that are well characterized there. And there's where you want, okay, now the hostorium of the parasite has invaded the host, and then the host mounts the successful defense response. And in these cases, it turns out to be something that would be familiar to your pathology listeners, right? Where you have an effective defense response. So there's some kind of a trigger that,
Like a hypersensitive
like a
response?
hypersensitive response, or something else, an enhanced lignification that the parasite can't get through. We don't have a whole lot of examples of this. There's just not that much good resistant germplasm out there. It's always been a limitation. As it's discovered and characterized, I think we'll come up with more and more mechanisms to describe it, try to figure out what it is that is an effective response against the parasite. And again, the parasite seems to be quite good at manipulating the hosts that it is adapted to. It's very effective at forming these hostorial connections and shutting down host defenses.
A question that I often ask when I'm studying insect parasites or fungi and plant pathogens is, do these parasites get their own parasites? And I'm wondering, do parasitic plants get parasites?
Not really. They have some things that will eat them. There's a few insects out there that will eat them. Some animals will eat them. They don't seem to attack each other. There's a few cases, I think, where they call it hyperparasitism, right? Where you have one parasite growing on another.
Yeah.
Growing on a mistletoe, a daughter wrapped around a mistletoe.
Right.
I haven't seen that. We tried to get, in my lab, we were growing daughter and broom rape at the same time. We were trying to get the daughter to wrap around the broom rape and form a connection that we could call successful. I tried, but I don't know that it was really successful in getting the nutrients from that parasite.
Now, in 2019, 2020, you were a Fulbright Scholar in France, correct?
Yeah.
What did you work on when you were there?
So I went to a laboratory there that the group I was working with works on sunflowers, right? Sunflowers is a big crop in France, oilseed crop. And there's this, great for us, parasite that attacks specifically sunflower. And it has a long history going back to the time of introduction of sunflower from the Americas to Europe, and then their big production in the former Soviet Union. And they had this type that's called sunflower broom rape, and they would breed a resistant version of sunflower, which would then be overcome by the parasite and so forth. A war of breeding and resistance overcoming those parasites, overcoming that resistance. So this sunflower broom rape has become quite a big problem in Europe in general. And a group there had sequenced the sunflower broom rape genome. And so I had a chance there to work on that and to interact with them as they were trying to describe this genome.
Yeah, thanks for that. Certainly can't ignore the fact that we're approaching the commercial holiday of Halloween, and there's some interesting Halloween-related questions I thought I'd ask you, or at least in the names of things. You've worked on Dodder, that's Scuta campestris. This parasitic plant has numerous nicknames, including wizard's net, strangleweed, devil's guts, devil's hair, beggarweed. What's your favorite common name for Dodder?
Oh, I don't know, but there's a group called they were calling it like a vampire plant, which I think is more appropriate for Halloween. I don't know if that's an official name. It like fangs to drink the blood of the host plant. I would offer that one
A
up.
vampire. Yeah, I like that one. Also, if we're on the topic of parasitic plants, generally, the famed corpse flower is a parasitic plant. Have you ever seen it in the wild?
No, are you talking about the Rafflesia?
Yes.
Yeah. I've not seen it in the wild. It's a fascinating plant because it lives most of its life within a vine in the jungles of the Pacific. And then it emerges into this huge flower that can be a meter across.
Yeah.
So it's one of the spectacular examples of strange plants in the plant kingdom. I would love to see it. I know that they're trying to grow it up in the U.S. Botanical Garden. I don't think anybody has produced it outside of its native range. But yeah, maybe it'll be coming to a place in the US soon. But it's interesting because it smells like you said a corpse plant, right? It smells like rotting flesh because it's attracting flies. But evidently, if you go looking for it, you have to be careful for the tigers that are also out there and attracted to it for the
Oh, wow.
yeah,
That's
that's dangerous. Like,
yeah. So Striga is commonly called witchweed. Why is that called witchweed?
I think because it bewitches the plant or something like that.
Okay.
Striga is interesting because it's a tiny little seed, and almost as soon as it makes that attachment to the host, that the host will stop growing. It's like way out of proportion to the impact of the parasite. And it's not that it is taking all that much resources, at least initially, but it must mess up the hormones or do something else to the host plant to really stop that shoot growth and then allow the parasites going to take over, basically use the root system for its own needs. It is a spell, it's cast some spell on the corn or something.
That's funny. It's not funny for the corn, but and it comes back to the main symptom of a lot of these interactions. It's a stunting of, and that's going to, the downstream impact is going to be reduced yield or no yield.
Yeah, yeah.
Now, daughter is in the morning glory family. We already discussed it. And what's really interesting is that the morning glories have fungal endophytes that produce some really interesting ergot alkaloids, these defensive compounds that some of them are real interesting from a chemical perspective. I was curious if daughter or other parasitic plants have endophytes, like fungal endophytes in them, or if anybody's ever done any like microbiome work on parasitic plants to see what's on and in them. Coming back to that, do parasites get parasites? But are there any other beneficial or asymptomatic passengers in those parasitic plants?
Yeah, that's a really good question. I don't know if I have a good answer for it. We have occasionally suspected that there might be other organisms, involved in there as part of helping out with them. I don't know, the parasitism or something, but I don't know any work. I haven't identified clearly endosymbiont or endophyte or anything like that in parasitic plants.
One of the things we had talked about earlier is the fact that there was this bidirectional movement at the hostorium. And I wondered, is there any clear examples of a plant virus going from the parasitic plant to the host? And were those viruses like symptomatic in both of the hosts essentially?
Yeah, so back in the 1940s, this was a classic test for a viral transmitted disease. You would get two plants, one with a disease, one without, and if you bridge them with a daughter, it will attack one plant at a time. So it'll make an attachment on your plant A, and then vine over, and you can make another attachment on plant B, and they're able to transmit viruses. And so that was a standard test for viruses at that time. And so that actually was kind of part of the reason that I thought that daughter would be a great one in which to study mobile RNAs, because it's transmitting viruses. It must be a form a good continuum.
Yeah, on that point, to serve as a conduit from one to the other is one thing. I wonder though if any of these viruses would have an impact on the parasitic plant themselves.
Yeah, and so that is something that it seems not to be the case. It seems that the parasite can convey the virus without being affected itself. Now, I don't know if that is strictly true, that the parasite has some ability to defend itself from the virus, if it just doesn't show symptoms the same way.
Because it
But
could be outwardly asymptomatic, but we know in parasitic plants that leaves turn into fleshy tooth-like scales. So how do you see a mottled leaf if you have just a scale that's not going to show that?
exactly. I like to think that the parasite's daughter has evolved a way to discern endogenous from external RNAs or viruses, things like that. And it has defense mechanisms to deal with it. But I'm not sure it's quite that sophisticated.
Yeah. One question I wanted to circle back to, and we were talking a little bit about this, it's about this aspect of host specificity in parasitic plant. So I know there's definitely parasitic plants that have a broad host range, and there's plenty that I know that have a very specific host. Is it more the case to be host specific, or do they tend to be like family specific? Tell me a little bit more about generally the host specificity of parasitic plants.
It's all over the map, but I think the general trend is as a parasite evolves toward being an obligate whole parasite, that they become more host specific. We tend to see the greater specificity. Usually it's for a range of plants within a family that it really does well on. But so for the witchweeds and broom rapes, you see a fairly narrow host range in those. But daughter, it's a obligate whole parasite. It has a very broad host. Oh, even within that, it has certain plants that it really does better on than others.
Yeah, I think about leafy mistletoes, like wherever a bird poops, like the seeds. Hey, I'll take this tree.
Yeah.
It's been wonderful talking with you today, and I've really enjoyed it. Thanks so much for your time.
Thank you. It's like being taken through an introductory lecture on parasitic plants by someone who really cares. So
We've just heard from Dr. Jim Westwood, Professor of Plant and Environmental Sciences in the School of Plant and Environmental Sciences at Virginia Polytechnic Institute and State University in Blacksburg, Virginia. As with all our episodes, be sure to check the show notes for links related to Dr. Westwood's research program. I'm Matt Kasson, host of Plantopia. Thanks for listening.